Um, so our agenda for tonight, we can go on to the next slide, please. We will be discussing highlights from NACFC. First, we're going to focus kind of on the care section, and we'll be talking about much of the new research that comes into caring with cystic fibrosis patients today. And especially, we know that that has been changing a lot recently, especially for patients that have access to CFTR modulator therapy like Trikafta. So we'll be talking about that a lot at the beginning, as well as infections and mental health and other areas that are of interest to the whole CF community. And then our second presentation will cover the latest findings in research and really what is on the horizon for new treatments that will be hopefully able to help all people with cystic fibrosis, regardless of what they have access to today. So with that, I'm going to introduce our first presenter, who is Dr. Raksha Jain. Dr. Jain is a pulmonary and critical care physician, an associate professor of medicine, and the director of the CF and bronchiectasis program at the University of Texas Southwestern Medical Center. She leads a number of clinical trials in CF and focuses her laboratory and clinical research on sex disparities and sexual and reproductive health in CF. So Raksha, thank you for joining us, and I will let you take it away.
Thank you, James, for that warm introduction, and thank you all for joining us this evening. We're really excited to have you with us this evening, and I'm thrilled to be sharing with you some of the highlights of the care and change in care that we learned at the recent North American CF meeting. These are my disclosures, most of which are related to participation in a variety of research studies. Okay, so just to start with, this was a slide that Elizabeth Tullis from Toronto presented at her plenary session on the third plenary at the North American meeting. And it really nicely highlights the growth in the CF population that is projected over the next many years. You can see in the purple graphic that this really represents the pediatric population and the total number of children we have with cystic fibrosis in North America. And you look at the blue color, which represents the total number of adults. Now, already this yellow line that you can see represents where we are today, and the adult population is already larger than the pediatric population, really thanks to a number of different things, including the care model, including airway clearance therapies, including lung transplantation, and a number of wonderful things that have already come about in CF care. And this future projection really overall shows that we expect that the pediatric size or number of children with CF will stay about the same, whereas the adult population we project to grow in the coming years to decades. And this is largely thought to be due to the future impacts of Trikafta and the fact that people are anticipated to live longer and healthier lives. So I think overall, this was a great message, and a message to the adult care teams in particular to prepare for the growth in the population, to adapt our model, and to make sure we're ready to continue to provide good care to people with CF, both adult and children. This slide was another slide presented at Elizabeth Tullis's plenary and provided by the CF Foundation registry team. And it really falls somewhat in line with the last slide that I just showed you. And it represents a graphic of, in the top row, children ages 6 to 8, and in the bottom row, adults ages 18 and older, where these circles are a distribution of the lung function represented by FEV1, where the purple color is people with more severe lung function, typically an FEV1 lower than 40%. And the light blue color is people who have really somewhat normal lung function, meaning greater than 90%. And you can see as you go from left to right, we're going from the 2000s to the middle panel, which is 2020, to the right-hand panels, which are 2040. And the real message here is that back in the 2000s or early 2000s, we had a greater percentage of the purple and yellow color, which represents people with moderate to severe, having moderate to severe lung function. You move into now, and those numbers have shrunk, both in pediatrics and adults. And then you move over into our projections of 2040, where in pediatrics, it's going to be less than 4% of people, or approximately 4% of pediatric people that will have lung function that's moderate to severe, and an estimated over 80% that will have normal lung function. The variety in the adult population down at the bottom right will be larger, but you still see that when you look from left to right, this lung function is overall improving in both the pediatric and adult population. So again, really news to celebrate overall. So now moving a little bit into some of the more recent studies that have been done in CF. And you can see that this is a presentation of the data from the CF Foundation sponsored PROMISE study that was led by Dave Nichols, Stephen Rowe, and Marty Solomon. And this was really a real world study that followed observationally people who started Trikafta. This baseline point you can see is pre-Trikafta, and the point to the right is 6 months after Trikafta. The left-hand graph represents lung function, and the right-hand graph represents the CFQR, or a symptom score that's often marked to evaluate respiratory symptoms in particular for these people. And when you look at what they saw in this study that really included over 400 people with CF, that even outside of a structured clinical trial, just looking observationally at how these people did, that when you just within 6 months of starting Trikafta, the change in lung function was almost 10%, a dramatic increase in lung function with Trikafta. Similarly, when you go over to the right-hand side, which represents the symptom score, you see an increase in the CFQR score, again, the symptom score, of over 20 points. And we really consider a 5-point change clinically meaningful. So 20 points is incredibly great to see. And on top of that, you see people with a score of 100, which is near normal, and 32% had a score that was near normal at 6 months. So again, wonderful news about the impacts for those on Trikafta in this study. Another real highlight study that was presented at the meeting this year was the Simplify study, another study funded by the CF Foundation that was really meant to look at withdrawal of medications after Trikafta. So I think as many of you may know, with the wonderful benefits of Trikafta, people have asked, can they potentially decrease the burden of their respiratory therapies, and will this impact their health? And this study, called the Simplify study, led by Alex Gifford, Nicole Mayer-Hamblett, and Dave Nichols, really set out to answer this with the help of the TDN. And what they did in this study is enroll people, and it was a study of over 600 people that enrolled in this study, and people were randomized to either continue or discontinue the hypertonic saline, or continue or discontinue their dornase alfa, otherwise known as Pulmozyme. And the goal of this study was to determine if discontinuing the therapy was going to be non-inferior to continuing, meaning will there be a change or a difference in lung function in particular if they stop their therapy. Importantly, this was a 6-week therapy, so people only came off of their therapy for 6 weeks. It was a randomized study, so in the hypertonic group and in the Pulmozyme group, people were randomized to either stay on the hypertonic or come off of it, or stay on the Pulmozyme or come off of it. And you can see here from the graphic on the left that amongst the people who were in the hypertonic arm, there was no difference seen, meaning discontinuing hypertonic saline was not inferior to continuing the hypertonic saline. Very similarly, when you look at the lower bar, that's the dornase alfa or Pulmozyme bar, that in those people, greater than almost 400 people essentially in that arm, that discontinuing the Pulmozyme versus continuing the Pulmozyme, there was no difference in the discontinuation of Pulmozyme. Now, it's important to recognize there's a few caveats and interpretations of this study that are very important to note. So first of all, this study recruited a fairly healthy population overall. In fact, the baseline or average lung function in the population in this trial was actually 97%, so quite high lung function. Additionally, it was a fairly short-term study, as I mentioned, 6 weeks, so we can't really glean longer-term effects from this trial, but nonetheless, it gives us some some hope that potentially decreasing the burden of therapy may be okay in certain people. But I think the investigators and all of us will acknowledge it's very important for you to have those discussions with your care team as an individual, because each person may have very different reasons why this is applicable to them versus not applicable to them. And I think the other important things to note is there are certainly more trials that are going to determine more information about this. There's an ongoing study called the HERO2 studies funded by the CF Foundation here in the United States that's really going to get information from a patient perspective on the implications of stopping some therapies. And there's another study based in Europe called the CF Storm study, which is going to be doing a very similar design as what you see here in the Simplify study, but actually following people up to 12 months. So I suspect that we'll have a lot more information coming in the subsequent years. So moving into some other highlights from NACFC. So this came from a session called Where Has All the Sputum Gone?, which I thought was a terrific session. And really, this graphic that you'll see here was provided by the CF Foundation Registry and presented in a talk by Dr. Patrick Flume from South Carolina. And what it represents is, historically, most people with CF have provided either a sputum culture or a throat or nasal swab at their clinic visits. And that's represented when you see on the very left-hand side in 2016, there's an adult bar and a pediatric bar in each of those. And you can see that the majority of adults would provide a sputum sample in clinic. Whereas in pediatrics, there's quite a few more that did not easily cough up sputum and in particular would already be getting swabbed for their culture, essentially. And this is so that care teams can detect if you grow different bacteria, such as Pseudomonas or staph in your cultures, and keep that in surveillance as needed when they need to treat you for different reasons. Then when you move along in this graphic to 2017, 18, 19, and most recently, 2021, you can see there's been fewer cultures captured overall. Some of this was impacted by the pandemic and in-person visits, and some of it has been that cultures have been more difficult to capture. And that's a real highlight that now not only do children have challenges in producing a sputum spontaneously, but adults too. A lot of people have produced very little sputum since the advent of Trikafta. And you look here that green bar, even in the adults and the kids, has grown, meaning there's more throat swabs occurring than spontaneous sputum production in clinic. So much of this session focused on understanding, does it matter what kind of collection we have? How do we figure out the right method? And much work is still ongoing, but preliminary data that was presented in this session did suggest that cough swabs may not be as good as sputum cultures in detecting bacteria. And there was a fair bit of consistent data that suggested that. Options that were proposed and are already being used in some places are sputum induction, where somebody can get a hypertonic saline induction, essentially a treatment to try to produce that sputum to get a better culture. And these are thought to show quite good results. In fact, results in this session that were comparable to a bronchoscopy, which is thought to be the gold standard, but an invasive procedure where you go down with a camera into the lungs, get a sample, and get a true sense of the bacteria in there. So I think the good news is there may be further options, and options your clinics may or may not already be doing, including the sputum induction. Other options that were brought up is that people are looking to potentially have mail-in samples. Now, all of this is still under study, and the CF Foundation has awarded a multi-center grant actually to assess the accuracy and reliability of these self-obtained respiratory cultures and see if this has potential to be adapted into the current care model. So pretty exciting work. Another session I would like to highlight is one that focused on non-tuberculous mycobacterial infections. So some of you may have heard of this, some of you may have had this experience yourself, but overall, the prevalence of these non-tuberculous mycobacterial infections in CF are thought to be about 13%, and it can vary from region to region quite a bit, but that's the prevalence in the overall population. The most common strains that we really face are this one that you see on the screen called Mycobacterium abscessus, and another one out there known as Mycobacterium avium. And these can be really challenging infections to treat. Sometimes they need to be treated, sometimes they don't. But when treatment is needed, it's quite difficult. It often requires multiple antibiotics, sometimes a combination of oral and IV antibiotics. And often these bacteria don't respond to these antibiotics because they have different ways to evade or work around our immune system. There's some ongoing studies by the CF Foundation, many of which are spearheaded out of National Jewish Health, called Patients, Predict, and Halt, which are already funded to understand more about the natural history, the strains that are occurring out there, the way that this spreads. But this was a really nice study that I wanted to share with you that was recently published in Cell out of Jerry Nick's group from National Jewish. And it shows a case of a 26-year-old man with Mycobacterium abscessus who'd been treated for over 4 years with a variety of antibiotics trying to get rid of this Mycobacterium abscessus. One of the other big challenges with this particular infection is it's often a contraindication to lung transplant because it can come back after lung transplant and be very vicious after the lung transplant. So there's a number of centers that won't transplant somebody who has this infection. So the team was trying to clear this infection because this person's lung function had been declining over time and he was nearing lung transplant. They really had not had great success. So they actually, using what's called a compassionate use study, where you get permission to try an experimental study on this person, gave them something called phage therapy. And you can see these little spider up here at the top. These are pictures of phage therapy. And what those are is they're actually viruses, sometimes naturally in the environment, sometimes engineered. And these little particles or viruses actually can selectively infect certain bacteria, get inside that bacteria and lyse it or break it open to cause that bacteria to die. In this particular case, the team used 2 engineered Mycobacterium phages along with continuing this person on their standard antibiotic therapy, and were actually able to eradicate or get rid of this Mycobacterium abscessus to allow this person to get a lung transplant. After this person did well, and as of yet, per this report, did not have that Mycobacterium return. So it's really exciting and gives us hope for the future of these types of studies and further use of this phage therapy. Then moving a little bit into well-being post-Trikafta. I think this has been something that so many people are discussing. I know many of you may have questions about this, and I know it comes up in my clinic all the time. And that since Trikafta's come out, there has certainly been an association with people describing an increase in anxiety, increase in depression, et cetera. And there's really much to be learned. It's not clear yet whether this is a direct effect or whether this is an association, but I think we all understand this needs to be studied more. So this was a really nice study by Erica Van Kitters at Dartmouth for a study that she's been funded by the CF Foundation to look at. And what she did here was actually survey a group of people with CF in the summer of 2022, and she surveyed over 1,000 people. And what she did was ask them a number of questions about their well-being. It included things such as their physical well-being, their experience of care, their social well-being, their daily life needs, and then their mental well-being. And each of these circles represents individual questions that were asked in the survey. And these arrows represent what were on the extremes of either positive, which are on the right, meaning things were better, or things that were worse, which ended up on the left side over there. So you can see, just walking you through this in the green, when it comes to physical well-being, the majority of these dots or questions, people answered positively, meaning they'd had a benefit since starting Trikafta. And you can see the one that's most extreme is cough, meaning that people had the greatest improvement in cough since the start of Trikafta. And then directing your attention to the bottom of that graph and these purple dots, this was mental well-being. And this was really the one category where the majority of the questions fell on the side of worse since modulator use, with anxiety being the worst of those. And, you know, I think it's important for us to recognize that there's been some wonderful benefits to well-being, but there is this aspect where people are reporting more anxiety and depression. And I assure you, I think we're aware of this. And I think many people who work in this space are working to understand how many of these factors are or are not related to Trikafta. What can we do about this? But I think it really highlights that this is there, people know, and it's being worked on. So I know currently there are people on different teams, that there's already been published case reports out there that people are trying dose adjustment of Trikafta to try to decrease some of these effects. And we also know that there's people who've chosen to come off their Trikafta because of this. So it's a high priority, and I hope we'll even learn more about this next year. Then moving into liver disease. So this is a session I was really excited because I admit that I do not spend as much of my time in liver disease as I would like to. But I think what's terrific news is there are some new liver disease guidelines that are coming out for the CF community. And this session really highlighted some of what these guidelines are going to be about. And I just wanted to point out a few of these, and this publication is still coming, but you can see what this graphic shows you is it's a graphic of screening for liver disease in all people with CF, not people just already diagnosed with liver disease or with ongoing abnormalities in their liver function testing. But these experts in liver care who are part of this guidelines committee essentially said that what we should be doing is getting annual labs on people. When it comes to liver function testing, we should be doing an annual abdominal physical exam, meaning examining the liver and spleen. So there's still a role for in-person visits that's very important. And we should be getting abdominal ultrasound every 2 years to evaluate the size and appearance of the liver and spleen. So I think that's something that we really are going to need to incorporate better into our care model. In addition, they have guidelines on monitoring for those who already have liver disease. And one of the real emphases or highlights was something called transient elastography or shear wave elastography, which are tests or non-invasive tests that assess liver stiffness along with labs for monitoring people who do have evidence of liver disease. And what these tests are is they use kind of an ultrasound type of probe that delivers low frequency pulses and is actually able to quantify the amount of fibrosis to the liver. Many of you may have already had one of these, but they're very quick and painless and really give hepatologists or GI doctors a good sense of what's going on in the liver. Another important thing to acknowledge is that these experts are also studying the impacts of Trikafta on liver disease and whether or not it can improve it or not, but I think the jury's still out on that, and I suspect we'll see that data coming in the near future. And it's also a component of the ongoing PROMISE study that's an observational study of people pre and post Trikafta use. So sticking with the GI theme, I think the other session that was really interesting to see was a session on the pancreas. It was actually called Putting the Pancreas on a Pedestal, and it really broke down different aspects of the pancreas to think about, particularly in the context of Trikafta. So you can see that there's two main functions of the pancreas. There's many others, but the main ones are the exocrine function of the pancreas, and that is the aspect where the pancreas secretes pancreatic enzymes. And we know that 95 to 98% of people with CF are pancreatic insufficient, meaning this process of secreting the enzyme is not adequate to release the enzymes needed for people to digest their food. So there's ongoing work to understand whether Trikafta can restore some of that enzyme secretion. And the challenge about this aspect of CF is really this dysfunction of the pancreas occurs in utero when people are developing and people are born with this pancreatic insufficiency. So thus far in the data they presented, really there's been very little change in the secretion of this pancreatic enzyme or restoration of this exocrine function in adults. That pancreatic insufficiency is potentially fixed or likely fixed, where it's not going to change in adults that had pre-existing scarring to their pancreas and lack of secretion. But they did show that in some younger people, and particularly younger people that do have some existing function to their pancreas, that there has been some evidence of Trikafta potentially improving that function. So that's exciting to see. And again, maybe even more exciting in the future years as Trikafta reaches younger and younger populations. The second component was the endocrine side of it, which is the side where the islet cells in the pancreas secrete insulin. We know that about 30% of people with CF have CF-related diabetes, and there's been a lot of questions about whether Trikafta can improve that or help that in any way. There again is a focus within the PROMISE study of understanding the impact of Trikafta on this diabetes. So far, this data that was presented by Andrea Kelly from UPenn at the meeting showed that the preliminary data so far shows that they're not really seeing changes in glucose tolerance tests. That's that oral glucose tolerance test many of you have taken. They're not really seeing changes in this, at least at the year mark. They're also not seeing an increase in overall insulin secretion, but they are seeing some changes in timing, including earlier insulin release. But they are seeing some subtle improvements in the hemoglobin A1C and even fasting glucose in some people. So this data is going to be continuing to be followed up to 54 months. And I think we'll see a lot more on this and understand it better as time goes along. So another area that I think is really exciting and something that has been an interest of mine and Dr. Taylor-Cousar's is the blossoming we have seen of people with CF having children. In particular, we've seen a number of mothers now and an expansion of the mothers with CF. And in fact, the number of pregnancies in women with CF has been rising over the last couple of years since the release of Trikafta. With that, there's been a lot of questions about the impact of Trikafta on the babies when these mothers continue it throughout their pregnancy. So this was a really nice report that Chris Fortner from Upstate Medical University presented at the meeting, where he actually showed a mother, a patient of his who was a mother with CF, who had one copy of Delta F508 and another CFTR mutation, and took Trikafta throughout her pregnancy. The father was a carrier of a CFTR mutation, a stop codon mutation, and the mother continued Trikafta during this pregnancy. She overall had an uncomplicated pregnancy and a very normal delivery overall. But very interesting, you'll see that the baby was evaluated by newborn screening, as all babies in the United States are now, to evaluate for CF. And typically, most states do something called the IRT, which is immunoreactive trypsinogen, and a test of whether the pancreas really is functioning or not. And this baby, who ended up with two CFTR mutations, the Delta F508 mutation and this E92X from dad, ended up with a normal immunoreactive trypsinogen. The baby also ended up with this FE, which is fecal elastase, which is another sign that the pancreas was working. That test was normal. The sweat test was 62, which is near normal. And the liver function testing was normal in this baby, and this baby had no meconium ileus. So very normal findings that would potentially have made this baby missed by newborn screening in many places in the US. The other incredibly exciting news was this was a baby boy, and the majority of males born with CF are infertile due to lack of the vas deferens, the tube that allows sperm to get out. And this baby at 8 weeks was found to have one palpable vas deferens. So this was really thrilling and a real indication that we have a whole nother area of science to study, and that there may be some ability to rescue some features of CF in utero. There's much more to understand about that and some ethical issues to consider, but there's actually an ongoing study sponsored by the CF Foundation called the Mayflower Study, which Dr. Taylor-Cousar and I are actually co-PIs on, and we're very excited to learn more about this in the next several years. So I don't want to forget the group of people who have already had a lung transplant with CF, and this is an area too that is being evaluated and not forgotten. And I think a couple of really nice highlights from the meeting is that we can see very clearly from registry data that Trikafta is delaying and even potentially preventing the need for lung transplant in many, many people. There's still a lot of questions out there, and I think one of the biggest ones that has come up is, what about the use of Trikafta post-lung transplantation? And in a nice study by the Lung Transplant Consortium that was presented by Kathy Ramos, she showed that when she looked at 15 lung transplant sites, that at this time, between 2019, October of 2019 and September of 2020, 91 people post-lung transplant had been started on Trikafta for a variety of reasons. They just observationally looked backwards at what occurred and how people did, and really found that about a third of these people, 39, stopped taking it, largely due to GI symptoms, but a number of other reasons. But several and many reported some pros to taking it. The biggest ones being improvement of sinus symptoms and sinus disease. Others had improvement in their BMI or improved malnutrition. Some reported improvement in their diabetes. And there's been some question about whether there can be improvement in chronic lung allograft dysfunction or a type of rejection of the lung. but there's many pitfalls that they saw and more to learn on drug interactions that it can occur with people on Trikafta who are also on immunosuppression medicines. Some potential increase in GI symptoms or reflux, gastroesophageal reflux disease, and excessive weight gain that was reported in many, and we've seen that in the general CF population as well. Increased liver tests, mood changes, as we already talked about, some issues with cost versus benefit, and then again, probably potentially no effect on donor lungs. So much more to be learned, but I think at least gives us a signal that people are trying that. And, I hope we can have some more answers to that in the future as well. So I just want to end with this last slide. I know I spent a lot of time today talking about the impacts of Trikafta, and I really just highlighted a few aspects of care from the meeting. But I don't want people to think that if you are not eligible for Trikafta or taking it for different reasons that you're forgotten, because I think we all understand how important that is. This was a really nice publication that was spearheaded by Emily's entourage and published earlier this year that was a survey for people who are not on CFTR modulators. They surveyed over 400 people, either with CF or their family members, to really understand the impact of not being on a CFTR modulator. And many reported that one of the most important impacted aspects of their life was their mental health. That was reported in almost 66% of these people. Others reported it in their physical health. Other people reported feeling isolated and witnessing the excitement or uplifting side of seeing their peers have access to modulators, but a little bit of disheartenment from not having access themselves. And you can see that in some of the quotes that came out of the study, including this one that says, "I'm glad so many have access to CFTR modulators, but at the same time, I'm concerned for my partner. I worry that 90% coverage would be seen as good enough. And I worry that the effect of being passed over by these incredible drugs has on my partner's well-being, both physically and mentally." And I will assure you that there are many, many more options being evaluated. And this is what's really going to be highlighted in Dr. Taylor-Cousar's talk next. And you're not forgotten. And I think everyone has their eye on helping every individual with CF. So with that, I really thank you for your time, and I'll turn it back over to James.
Thank you so much, Raksha. That was a lot of really interesting information. And that's really just the highlights of what has been talked about in current day CF care at the North American CF Conference. So thank you for that presentation. And now we're going to switch over to Dr. Jennifer Taylor-Cousar, who is going to be talking more about the future of CF research and what is exciting on the horizon. Dr. Taylor-Cousar is a professor of internal medicine and pediatrics at National Jewish Health, where she is the co-director of the Adult CF program and director of the CF Therapeutics Development Center. She has been on a national and international primary investigator on a number of clinical trials, and her expertise in clinical trial design and conduct and therapeutic development for the treatment of CF lung disease. So thank you for being with us today, Jen, and I will turn it over to you.
Hey, thanks very much, James, for that introduction, and thanks to the organizers for putting this together and all of you for spending part of your evening with us. So these are my disclosures. Like Dr. Jain, most of mine are related to the fact that I'm the primary investigator on a number of our studies here at National Jewish. So I really wanted to make sure that we started out on the same page about how we think about the strategies to treat people with cystic fibrosis. So on the left side of this slide are those things that we consider to be part of the path to a cure. So those that impact DNA or gene therapy, so the blueprint that tells the cell how to make the CFTR protein, the chloride channel. RNA, which is the message, the copy of the DNA that has really specific instructions for the cell on how to make the protein. Then there's the protein itself, and you heard a lot from Dr. Jain about the modulators like Trikafta that we use to treat the protein. And then there are the things that we use to treat the signs and symptoms of CF, sort of like she mentioned with dornase alfa or Pulmozyme and hypertonic saline. We're really going to focus over the next few minutes on the first three aspects of care. So based on the 6-month randomized trials that tested Trikafta in adults and older kids, and then later in kids that were 6 to 11, and then even a little bit later, work done in cells for those who have rare mutations, that really led to the fact that at this point, the majority of people with CF are mutation eligible for highly effective modulator therapy. So highly effective meaning that it improves lung function by a lot, as you saw in Dr. Jain's presentation. It improves body mass index, it improves people's quality of life, and it decreases their pulmonary exacerbations. And what we've seen is in the past is that really short-term data. But one question that we ask ourselves as clinicians and as researchers is, will those really rapid gains that occurred in the trials be sustained over time? So this first graph that I'm showing you was presented in a workshop as well as in plenary 2 by Dr. Polanyi. And it's showing you on the Y, or the vertical axis, the absolute change in percent predicted FEV1, so lung function. And then on the x-axis is the time in the study. If you focus on the left side, that's the portion of the trial where people were randomized. So they either got drug or they got placebo. And you can see that those who got drug right away had that rapid improvement. Those who were on placebo had a little bit of a decline. But then once people were offered open label drugs, so everybody was getting drug, people sustained their lung function, not just over a short period of time, but all the way out to week 144, so 3 years. So that's very promising. And it tells us that this drug is continuing to work for those who can take it. Now, I told you that this was in older kids and adults, and then we got approval down to 6 to 11, but we really think it's important that we treat people as early as possible. And you saw how exciting it was when a baby was treated while it was in its mom's womb, and it actually had prevention of some of the complications that we see in CF. So this study, also presented by Dr. Polanyi in plenary 2, was looking at Trikafta for 24 weeks or 6 months in kids who are 2 to 5 years old and have at least one copy of F508del, the most common mutation. And what you can see, if you focus first on the top of the slide, is that people's sweat chloride went down, and it went down within that first 4 weeks. So in these children, their sweat decreased by about 60 millimoles per liter, so sometimes going into the normal or indeterminate range. And that was sustained over the first 6 months of the trial. Also, if you focus on the bottom part of the graph, it's really hard to do the spirometry that we do in older kids and adults in little kids. They can't follow that instruction to blow out really hard for a prolonged period of time. So instead, what we do is measure something called breath washout, where there's a tracer gas, and we measure how long it takes for that gas to go out of the lungs. So you actually want to see a decrease, and that means improvement. And you can see that in these little kids that were 2 to 5 years old, there was a decrease right away in that first 4 weeks, and that was sustained over time. We're hoping that this data will lead to the approval of Trikafta in 2 to 5 year olds sometime next year. So where are we with the CFTR modulator landscape? So based on the US patient registry, up to 94% of people with CF could ultimately benefit from highly effective modulator therapy like Trikafta. But there are other modulators that are being studied, and I'll talk about one of those momentarily. Of course, as Dr. Jain mentioned at the end of her presentation, there are people who will not be eligible based on their mutations, and there are some people who cannot take Trikafta because of the side effects. And in the later part of this presentation, I'll address therapies that we're looking at for that group of people. So first, let's talk about this other modulator that's being tested right now in cells. So Sionna has tested their modulators in human bronchial epithelial cells, so cells from humans that have 2 copies of F508del. And in this first graph, we're looking at chloride transport on the vertical axis, and then the different therapies that were used in the cells on the x-axis. Green is the amount of chloride transport that you would expect to see in cells that don't have CFTR mutations, in other words, wild type cells. So when there was control given, there's no change in chloride transport. When Alex-Tez-Iva was given, there was about a 60% improvement, but when this drug called Scion 638 was added to Trikafta in these cells, you can see that the chloride transport got all the way up to wild type levels, so levels of cells without CFTR mutations. So very exciting. The other thing that they looked at was, what about if you use their triple combination of drugs in human bronchial epithelial cells? And I've highlighted for you what happened with Trikafta when Trikafta was given to those cells, and then what happened when the Sionna triple was given to the cells. And you can see again that while Trikafta got those cells up to about 60% of chloride transport, and you gave the Sionna triple, it went all the way into that wild type or range for cells without CFTR mutations. So they're hoping actually that this drug will be tested in the clinic in a clinical trial in the first half of 2023. So now let's switch to talking about genetic therapies. So again, DNA is the blueprint for the cell to make the CFTR protein. The RNA, the mRNA, is the copy that has really specific instructions for making that protein. And what we want to do with genetic therapies is reprogram the cell to make a functional CFTR protein by providing the correct instructions. So we need to find a way to get it into the cell, and then we need that message to get translated into the correct protein. And again, I want to make sure that we're all on the same page when we talk about genetic therapies. So I borrowed this slide from Martin Mentz and Hilary Valerie, and they presented it, I think, in 2019 at NACFC. But they used the example of, what if CFTR were a car? Let's think about how we might fix the car when there are car problems. So with mRNA, it's very short-lived. So if we use an mRNA therapy, it's like a rental car. So you have to replace it, and you'll have to do it several times over and over again because it lives for a very short period of time. With gene therapy, you're replacing the whole gene. So that's like getting a brand new car. And then gene editing, which is a little bit further away from being in clinical trials, that's actually going in and fixing the problem in that particular person's cells. So repairing the car. And again, that one's a little bit further away, so I'm going to focus for the next few slides on mRNA therapy and on gene therapy. So in plenary 1, Dr. Rebecca Dara actually presented the data from 2 studies of mRNA. And with the mRNA therapies, they're using lipid nanoparticles, and she referred to that as a fatty ball. So the mRNA is very fragile, it's short-lived, and so you put it in this protective fatty ball to help it get into the cells where it needs to go so that you can actually fix the mRNA, give it the correct message. So Arcturus shared their data for their lipid nanoparticle, LUNAR, and they actually administered their drug to CF ferrets. So they had the ferrets inhale the drug, and then they looked in the cells to see if they saw that corrected product there, because there are 2 ways we usually think about looking at whether or not a genetic therapy is working. One of those ways is, do we see the corrected product in the cells? And the other is, does the chloride transport get better in those cells after we deliver the product? So you're looking at the trachea and the bronchus. So trachea, the big tube in your neck that goes into your lungs, and then a bronchus, it's one of those branches that goes off of the trachea. And what you see is green. That's what you're looking for. And the reason we're looking for green is because when that mRNA product was added into the nebulizer, there was also something called recombinase put in it. And what that does is, when you actually get the product to where it's supposed to go, it turns the cells from red to green. And you can see a lot of green in the airway. And what's really important is that even though there's this big glob of mucus, which we know can get in the way of these therapies getting to the right place, even though that glob of mucus was there, you could actually still see the green in the cells, again, showing that the product got to where it was supposed to go. Another company that shared their data is Recode. They are also using a lipid nanoparticle to deliver mRNA, the correct message. And what they did was nebulize their product onto cells, again, human bronchial epithelial cells, via a mesh nebulizer. So sort of like you would take a nebulizer with your dornase alfa or your hypertonic saline, they used something to nebulize onto the cells. They then looked to see if chloride transport got better after the cells were treated. So if you first focus on the left side, they looked at cells that had 2 copies of F508del. And on the y-axis is again chloride transport. And you can see that although the mRNA did not cause the chloride transport to go up quite as high as Trikafta, it did get it to the levels of Orkambi, which we know stabilized a lot of people with CF. Similarly, when they looked in cells that had 1 copy of F508del and a G542X mutation, a nonsense mutation, although they didn't get chloride transport to Trikafta levels, they were able to get chloride transport in these cells to Orkambi levels, which again we think would be a great start for people who currently don't have any therapies that work for their mutations. So really, they're trying hard to make this product work as well as possible, and they're hoping that in the next year they'll be able to move this into clinical trials. So that was mRNA therapy. There was also data presented at the meeting on gene therapy, again, replacing the gene. And this was presented in Symposium 1 by myself for 4DMT, as well as Dr. Eric Alton and Dr. Peter Walken. If you'd like to watch that, that symposium is on YouTube right now. But I'm going to focus, just for the sake of time, on the 4D data. So this is a adenovirus vector that's been modified. In the past, so when the gene was discovered back in 1989, we thought gene therapy would be around the corner. And the first thing they tried to use was an adenovirus vector to carry that corrected message into the cell. The problem that they ran into was that, one, there is a lot of thick, sticky mucus that you have to get past in the lungs of people with CF, but also the body's natural defense to a virus is to try to fight it off with inflammation. And so they really just weren't able to get enough of the corrected gene into the cells. So 4D sort of thought of this the way we think about high throughput screening for modulators. They looked at billions of ways of getting the corrected gene into the cell. And they tested them over and over again until they came up with one that worked really well, called A101. And what they showed in an animal model was that this A101 vector or carrier was able to get past those barriers, so past the thick mucus and past the body's natural defenses of antibodies and something called defensins. So based on that really promising preclinical data in animal models, it was moved into a phase 1 clinical trial. So 3 adults with cystic fibrosis were given 1 dose of 4D7T by aerosol, by a nebulizer, and it was a low dose because it was the very first time in humans that we were doing this study. We showed that it was safe and well tolerated in those 3 people with CF. And when we did biopsies of their airways, 100% of the lung samples actually showed that we got the corrected gene into the lungs. And if you look there on the right, there are 2 different pictures of cells in the lungs. First is a 40x magnification, and the bottom is an 80x, just to look at it a little bit closer. And all the areas where you see the brown staining are areas where we got the corrected gene into the lung. And this is the very first time since gene therapy trials started in people with CF that we were able to show that we could get the corrected gene into the lung in the lower airway, because in the past they only showed it in cells from the nose. So very, very exciting, but again, just the first 3 people with CF who've been studied. We're now moving into a second cohort where people will get the higher dose, and we'll look again to see how much of the corrected gene got into the airway, but we'll also start to look at lung function and quality of life. So I think that gene therapy seemed very promising a long time ago, and then we hit some bumps in the road, but now we're really on a good path to finding therapies that we think are going to help people with cystic fibrosis. So I'll stop there and I'll turn it back over to James, because I think we have time for just a few questions.
Thank you so much, Dr. Taylor-Cousar. So we are going to take a few questions that we have put in the Q&A. So Jen, I'll start with you. You mentioned the Sionna study, and the question was, is this a different formulation of Trikafta's VX121 study? So can you kind of explain the differences between those?
That's a great question. And actually, all of the Sionna drugs are completely different than the ones that are in Trikafta. So it's a whole new series of drugs that are modulators. They all work a little bit differently.
Great. And with the mRNA therapies, do we know about how close those are to phase 1, 2, and 3 clinical trials?
That's a great question. Actually, we are going to start those in 2023, the mRNA trials.
That's awesome. And which phase is that?
It'll be phase 1, because these are the first studies in people with these drugs.
Great. Now I'll have one for Raksha. In the care area, are people able to take Paxlovid if they get COVID and they're also taking Trikafta? What are your clinical considerations there?
Sure, that's a great question. And I didn't touch much on COVID in my slides, but it was definitely highlighted at a few sessions at the North American meeting. Overall, so there is a drug interaction between Paxlovid and Trikafta. Paxlovid is a very effective antiviral that has been made available for treatment of COVID, but there actually has been some work to look at a method of dose adjusting the Trikafta so people can take Paxlovid. And, I think it is something to the effect of, on the first day, you take the Paxlovid, you take 2 of the orange capsules of the Trikafta, then you have to hold your Trikafta for the subsequent 4 days. And then I think you take another dose of just the orange tablets and hold it again. But I know the care teams have been given this suggestion on the adjustment. Now, there is another antiviral out there that does not have the same interaction with Trikafta, and there's also monoclonal antibodies available. But, for sure in our own clinic, we have dose adjusted Trikafta and given Paxlovid to people who needed it before, so it can be done.
That's great, and definitely sounds like it's an important thing to talk to your CF team about if you find yourself in that situation. And also, can you clarify for us, we had a question that said, with the Simplify study, 6 weeks doesn't seem like very long to stop nebulizer treatment. So can you remind us how we're studying this going forward?
Sure. So first of all, I think the investigators and study team of the Simplify did review data out there, and it has shown that 6 weeks is adequate to understand if there's going to be a change in lung function with stopping a daily therapy. So that was one of the reasons that they chose the 6-week time point. But at the same time, I think we recognize that the long-term impact of studying that daily therapy can't be gleaned from this 6-week study. So there's a couple of ongoing studies. One is called the CF STORM study being led in Europe that's going to do a very similar design, but follow people for 12 months. And then there is a study here in the United States called the HERO study that is really going to look at it in a more real world patient perspective fashion that's also going to be long term. So I think we'll have a number of other ways to validate that over a longer period of time.
That's great. So I'll take this one back to Jen. Why do we use viruses to deliver genetic therapies?
Yeah, that's a great question. So there are a couple of different ways that I mentioned, because the problem is you have to have something to get it into the cell. And one of the things about cells is that they're made to keep things out. And so viruses actually have evolved to be able to get into cells, especially for the case of CF, you want to get into respiratory cells. So the reason that we use these modified viruses that we don't think will make people sick, that was like one of the ways that they were modified, is because there's already a receptor, or basically a key, to get in the door of the cell for these viruses.
Great. Let's see which one I want to do next. What about, people may have heard about CRISPR in terms of gene editing? Is that something that could be used in CF someday?
So we do hope, I mean, we really think that's probably the way to cure CF for every single person, is to be able to edit that specific mutation that they have. But as I mentioned earlier, it's probably a little bit far away from clinical trials at this point, because it's complicated to make sure that we're getting it exactly where it's supposed to go and not clipping out parts that we actually really need to be there. But we do think it'll be a viable option for CF at some point. And even if you've taken CFTR modulators before, because again, CFTR modulators are affecting the protein, not the actual gene or the mRNA. So even if you've taken a modulator, you'd be able to be helped by these therapies.
And speaking of modulators, you showed a slide comparing Trikafta to the Sionna modulators that are in development. And we can see that they sometimes have different effects on sweat chloride. Do we think that that would translate into a better clinical effect in patients?
Yeah, that's another great question. So one of the things that's become very clear over time with the modulators is that if you look in human bronchial epithelial cells, so cells from people, the response in those cells in the petri dish translates very well into what happens in the clinic. So if you look, for example, at how or can be compared to Trikafta, we saw in the cells that the response was lower with Orkambi than it was with Trikafta. And so then when we got to the clinic, we saw that there was a much higher response. So there is a very good correlation when you look at human bronchial epithelial cell response to what will happen in the clinic. So we are optimistic that if those Sionna modulators make it into clinical trials as expected later this year, that they have the potential to have even more effect on the clinical picture than the modulators that we have approved right now.
And it also sounds like it would be very important for patients and clinicians to be able to have more options for modulator therapy as well. So now I will give a question for Raksha, which is, are we learning anything about if Trikafta affects hormones in specifically in women? Is the question that was asked, but I think it's interesting to ask that more generally for everyone.
Yeah, that's a great question. I mean, as far as at least I'll start with drug interactions with hormones first off. And as far as we know, Trikafta does not interact with hormones that are taken supplementally, particularly contraceptive therapy. Of the modulators that were out there, actually, there was a drug interaction with Orkambi that made people have to use a secondary form of contraception, but there is not that drug interaction with Trikafta. And really, when you look at, we don't know the big answer to that question of, is there a change in hormones pre-post Trikafta? We don't yet believe that there's an interaction with Trikafta and hormones, but I think what we have to understand is potentially as an indirect effect with improved health, et cetera, could we see a change in actual hormone levels? And I think particularly in men, there have been reports of hypogonadism and low testosterone in men for reasons that aren't quite all clear, but it's possible that we may see an improvement in that over time, but we really don't know yet. And I think that's an area that is definitely ongoing. And in fact, I have some funding from the CF Foundation to look at that question using some PROMIS data. So I think we're hoping to find that out, but I certainly would not dismiss the fact that there are people reporting day-to-day changes in menstrual cycles and different aspects of their health. And why that is, I can't say that we know yet, but I think hopefully, again, we'll find out in the near future.
Great. So I'm going to ask two more questions. This next one will be for Jen. And speaking of stop mutations, we know one of the ways that those might be able to be treated are often called read-through agents. Can you tell us what's happening in the CF research world, specifically for people with those stop mutations that often end with an X?
Yeah, that's a great question. And we actually really hope that the mRNA therapies will be able to help those people who have those mutations. But in terms of your specific question about read-through agents, unfortunately, we received news earlier this year from the company called Elox that was looking at a read-through agent. And although in cells they had shown that their drug actually worked, and it worked even better if you gave ivacaftor in addition to the Elox drug, in the clinical trial, it did not make people's lung function better or their chloride transport better. We think that is probably because of the way it was being given. So it was being given as a subcutaneous under the skin injection. And so we just don't think enough was getting into the cells. So what they're trying to do is reformulate that drug so that it can be given by inhalation. So we are hoping that that's going to happen. We are not yet sure if it actually will. But again, if the mRNA therapies and the gene therapies work, those would work for people with nonsense mutations.
All right. And for our last question, I'm going to ask this to both of you, since you are both CF physicians and researchers. And we have seen with Trikafta, enormous improvements for many people with CF. And there's also a set of people with CF that don't have access to these therapies yet. And if you can, for a minute, talk a little bit about, as a clinician and a researcher, how you kind of balance these two very diverging patient populations that are starting to have really different needs. So how about Raksha first?
Sure. Thanks, James. That's a really good question and a really hard question. I mean, I think every person is individual. So I'd say, first of all, I think that for me, I don't have one recipe of care for every person. It's very individualized already. And I think the biggest difference is maybe I'm trying to keep the people who don't have access to modulators motivated and hopeful, because I think there is still, as Dr. Taylor-Cousar described very nicely, so many wonderful things on the horizon, and just keeping them in a state of not giving up and working hard, et cetera. But I will also say that there's plenty of people on modulators that are still struggling and maybe have not had the level of benefit they hoped, are still getting sick in different ways, and maybe not, seeing what everyone else is seeing. So I think it's very individualized. And, for some of those people, we may in the future be able to adapt care in different ways to, again, potentially make it less burdensome, maybe make fewer visits a possibility, et cetera. And for some, we may have to stay quite aggressive and quite in tune with their care. So I think it's really, knowing each person and them talking to their care team and coming up with an individual plan.
Great. And Jen?
Yeah, I think that Raksha stated it really well. I think one of the things that those of us who went into CF love about what we do is that we develop long-term relationships with people so that you can individualize your treatment for them and what they're able to do, what they're willing to do. And the other thing I think that's really important that Raksha addressed is that, at every single clinic visit, I talk to people about the research that's going on, whether they're on Trikafta or not on Trikafta, because again, there's a spectrum of response to Trikafta, there are side effects. And so I think it's really critical that people know what is coming in the future, in the very near future, in the more distant future, because I think it does give people hope.
That's great. Thank you so much, Dr. Jennifer Taylor-Cousar and Dr. Raksha Jain, both for your presentations and summarizing what we've seen at the North American Cystic Fibrosis Conference this year, and also for all of your work as researchers and clinicians. So with that, I will close our webinar. Thank you very much, everyone, for joining us. Thank you for submitting your questions, and I'll hope to see you next time. Good night.
Thanks, James, for a great moderated session.